Lab 6. Fermentation and respiration. After you study this laboratory, you should be able to 1. Understand the overall balance equations for fermentation and aerobic respiration. 2. Distinguish between the products and efficiency of fermentation and aerobic respiration. First, let's talk about respiration. Respiration is the process in which nutrients are converted into useful energy in cell. There are two processes. 1. Anaerobic respiration and 2. Cellular respiration. Anaerobics is respiration process without oxygen. This process also often called as fermentation. Fermentation is an incomplete breakdown of glucose. As you see here, it starts from glucose molecule, and then the first process is glycolysis. When in a condition without oxygen, then the fermentation is taking place. It depends on organism. The product of anaerobic respiration can be either lactic acid or the ethanol. As I say, anaerobic respiration is really depend on organism. Different organism will provide different product from fermentation. For example, Saccharomyces provide carbon dioxide and ethanol. That's why we've been using them for wine and beer industry. Or another example, Bacillus, Lactobacillus, and Streptococcus. This group of organisms provide lactic acid. That's why it's been using to make cheese, yogurt, and soy sauce. Another example of an aerobic respiration that we are familiar with is yeast fermentation, right? That been used in wine and beer industry and even make bread. The process of this, besides provide alcohol, is also have another product which is carbon dioxide. This is the reason why we see the bubble in wine and beers, and also is responsible for rising of bread during fermentation, and that's why after we bake them, you see the cellular respiration. This one is different from anaerobic respiration because this process is complete convert glucose into inorganic molecule and produce a large amount of energy. That's why I talk about 36 to 38% ATP. Similar to fermentation, the aerobic respiration also starts with glycolysis. Glycolysis is basically convert glucose into pyruvic acid, which is doing convert from glucose to pyruvic acid, which is received from glycolysis, then enter citric acid cycle, or we are known as Krebs cycle. From Krebs cycle, we will receive carbon dioxide, FADH2, and NADH. Those NADH and FADH2, which is received from Krebs cycle, then going into electron transport chain. In this process, will make 32 to 36 ATP. Exercise 1. Yeast fermentation. Each person will obtain one flask. Our experiment will design composed of 4 conditions. By what you will do, you have to put 100 ml of different carbon salts, which are glucose, sucrose, 10% starch, and deionized water into each flask. And then add 1 gram of dry baker yeast into each flask. After that, quickly place the balloon over the mouth of the flask. Incubate flask at 30 degrees Celsius and observe every 15 minutes.
Remove the balloon. Then take 1 ml of the mixture from each experiment set to test with 1 ml of phenol. Red solution. Examine the color and measure the pH with pH indicator paper. Exercise 6.2. Phenol red solution as pH indicator. Examine the color of the indicator before starting the experiments. Then complete the paragraph.
First, we will test the pH of the indicator changing by adding acid and basic solution into phenol red indicator solution and then measure the pH by using a pH paper. We will test acid first. This is the result after add acid into phenol red solution. Next is basic solution. This is the result after adding base or basic solution into phenol red. And next we will test the pH by using a pH indicator paper. We need to submerge this paper into the solution and then let all 4 color band in that paper, right, get the solution diffusion and let it dry for 1 to 5 minutes. After that, compare the color of these 4 color bands with the indicator on the box. Let's see what is the pH for the acid solution when adding into phenol red. So we need to match all 4 bands. And then after that, the basic solution. So it does not match from pH 0 to 7, so we change to another size of the box, that is the range of pH 7 to 14. It's time for you to write a report.